International Convention on Green Environment Technology Entrepreneurship through Innovation. In this conference, entitled Sustainable Development in Allied Science, Engineering and Technology, we have tried to bring together scientists, researchers, industrialists, academicians, students, entrepreneurs who are working in different areas of sustainability to share their experience and exchange and cross-fertilize their innovative ideas. I would like to welcome Dr. Arpit Srivastava for the welcome address. Dr. Arpit Srivastava is currently working as Assistant Professor at Amity Institute of Biotechnology, Amity University, Rajasthan. He has been awarded the ICAR SRF Fellowship in 2015, securing an impressive 5th rank in the general category nationwide. He completed his PhD in Food Science and Technology from Banaras Hindu University in the year 2021. He possesses a strong background in both academic and industry settings. He has held positions as Assistant Professor at Ganpat University and AKS University prior to working here. He has also worked as Senior Research Fellow at Central Institute of Agricultural Engineering in Bhopal. Prior to his academic career, he gained valuable industry experience as a technical officer at Sanchi Dairy Plant for 2 years and a quality executive at Coca-Cola Aurangabad for 1 year. He has published over 20 research and review articles in various prestigious international and national journals, along with 10 book chapters. His area of specialization is food processing and nano encapsulation. I would like to welcome Dr. Arpit Srivastava for the welcome address. Sir, please.
Okay, thank you, Manishita ma'am, for the profile and for introducing me. And now I would like to welcome you all who is present here and online on this distinguished gathering. And we are honored to have among us Dr. Swapnil Tijgule, and who has made significant contribution in the area of bioinformatics. Dr. Swapnil Tijgule. Yes.
Hello, Arpit.
He's a postdoctoral researcher and computational biology from June 2022 to October 2023 in Walter Eliza Hall Institute of Medical Research, Melbourne. He has done MTech in Information Technology from the August 2013 to July 2015 from Indian Institute of Information Technology. And he has been nominated for the prestigious Shemin Science Fellowship for the University of Melbourne and second prize in the e-poster competition at the AMCI Bioinfo Summer Hybrid Conference. So I welcome Dr. Swapnil Tijgule. Thank you.
Hi, Arpit. Can you hear me?
Yes, we can hear you, sir.
Okay, I'll just share my screen.
Yes, sure, sir. We are just unsharing this from our end so that you can share from your end.
Okay.
Yes, sir. You can share. Please, sir.
Can you see my screen?
Yes, sir. Yes, absolutely perfect. We are ready to go, sir.
Okay, yeah. Thank you, Arpit, for the kind introduction.
Okay. So 1 second, please. No.
Yeah, now it is visible.
Yes, sir. Now we are ready to start. Thank you, sir.
Okay, thank you. Hi, everyone. Yeah, thank you, Arpit, for your kind introduction, and And I would like to congratulate the committee for organizing this wonderful conference, and also I'd like to thank you for providing me this platform to present my work here. So I hope everyone is enjoying this conference so far, and I hope this is the last day of the conference, and you might have got a lot of insights from different talks and all, and got new opportunity to network and collaborate. So that's the main purpose of attending the conference. Yeah. So a little bit of myself. I'm a postdoctoral fellow. I'm currently working at Icahn School of Medicine at Mount Sinai here in New York. So here I'm working on the genomics of neurodegenerative and neuropsychiatric disorders. I've joined here recently in this year. So currently I'm currently still developing the projects in this field. But today I'll be presenting my work from my previous affiliation, which I conducted this research in Walter Eliza Hall Institute based in Melbourne, Australia, where I used to work in genomics of infectious diseases. So this is, I would like to just show you my workplace and everything where I currently work. So this is the Mount Sinai Hospital, and this is the very beautiful view of New York where we can see from our office. So it's really pleasant working here. And this is my previous affiliation, Walter Eliza Hall Institute, where I conducted most of my research, where I finished my PhD and also did a 1 year of postdoc here. So the work I'll be presenting today is based in this research, which is based in Melbourne, Australia. So coming back to the topic, which I'll be today presenting on the applications of evolutionary genomics in infectious disease. Just to give you a brief overview of evolutionary genomics, so it's a field of research where we try to understand how genome within an organism evolves so that it helps them to adapt or diversify within an ecosystem. Because this evolution is very necessary for any pathogen to survive in any given condition. And we try to understand this genome evolution by studying the genetic exchange happening within the genome. Because this genetic exchange might play a different role, and we try to understand the impact of these genetic changes on the organism's evolution and its adaptation. In terms of infectious diseases, this is very important to understand the evolutionary genomics of a pathogen, because this helps us to understand how a pathogen can emerge into a new field, how it can adapt to different hosts, and also transmit between different host conditions. This information in infectious disease is very important to develop any preventive and control strategies to stop the disease transmission and combat against the infectious diseases. So today I'll be focusing on this one of the very important infectious disease, which is diarrhea. Diarrhea is the second leading or maybe the most important infectious disease and which causes a high disease burden, particularly in the young children below the age of 5 years. Particularly the children that are under malnutrition, or also the children which have very low immunity or low immune response, those children are highly susceptible to this diarrheal infection. Because once they have this particular infection, then they have these long-term effects, and they can also have effects in their cognitive and brain developments. That's why it's very important in young children. Despite this significant impact on human health, there is still lack of funding into this research. So that's why here currently we are focusing into this diarrheal disease. So just to focus on any disease, we first need to understand what are the major pathogens or major infectious agents that are causing this particular disease. There are a wide number of organisms that can cause diarrheal disease, including viruses, bacteria, fungi, and different kinds of parasites. So top leading cause of diarrheal agent is a rotavirus. So since it's a virus, so already a lot of development and research has been made into that field, and a lot of programs has been developed to stop the disease transmission caused by this rotavirus. So therefore, here we are focusing on the second topmost leading cause of diarrheal disease, which is a parasite, Cryptosporidium. The infection caused by this Cryptosporidium is known as cryptosporidiasis, and every year it leads to around 200,000 deaths and 4 million cases each year. Most importantly, to date, there are no effective treatments available against this parasite. And very interestingly, this parasite is resistant to water treatments. So that's why it's mostly important. It's mostly very important pathogen that can cause a waterborne outbreaks. So all over the world, more than 50% of waterborne outbreaks has been caused by this parasite. This parasite can be transmitted to either food, water, or can infect to humans and also animals. So there's a wide range of scope through which this parasite can infect. This Cryptosporidium parasite came into limelight in 1993, where it almost infected the entire city. The city is Milwaukee in USA, where it infected around 400,000 individuals, and it costed more than $500 million to recover from this infection. Cryptosporidium is a very complex organism, as it has multiple species, and with all those species have these multiple subtypes. So for example, in simple terms, I can say that, for example, in SARS-CoV-2, which causes this COVID-19, so you might have heard this COVID-19 has different substrains like Omicron, Alpha, Delta, and all those substrains have different variants such as BA4, BA3, and like that. So like SARS-CoV is very complex. So like Cryptosporidium is also the similar organism, which has a lot of multiple species and multiple subtypes. But among these multiple species, the two species are very dominant and very common in human infections, which are Cryptosporidium hominis and Cryptosporidium parvum. These two species have been commonly identified throughout the world and identified in many of the countries, even developing and developed countries. But the most of the studies in this research is mostly limited to the genotyping. Genotyping is basically means that identifying which particular species is present in which geographical region, or in which in which kind of animal or which kind of host it's been detected. And this is mostly done by using a single marker or a combination of multiple markers. So here are the few examples where how people have used this genotyping approach to identify or detect this particular kind of a specific species or specific subtype in different animals and in different geographical regions. Since most of these researchers or most of these studies are only dedicated to genotyping studies, there's a lack of whole genome studies, because we still lack the entire picture, because most of the people or most of the researchers are only focused on single gene or just combination of multiple genes. But in the recently 2 or 3 years, there are some new publications are coming, or many people are taking into interest, which are conducting whole genome studies. But those studies are either restricted to small geographical region or to a single host. And also they have a very limited sample size. So still, we are left with the very important questions: how this particular pathogen, Cryptosporidium, evolves, how it adapts to its host, and also how it's transmitted between different hosts, and what are the main virulent factors, the virulent genes, which is very important in this pathogen, and they use them to infect to the host and goes and attaches to the host and leads to the infection. So therefore, in my studies, we designed 2 different kind of studies to understand the evolutionary genomics of 2 dominant species, which is Cryptosporidium hominis and Cryptosporidium parvum. So in short term, I'll be just calling them C. hominis and C. parvum. So in study 1, we focused on this anthropomorphic C. hominis. So anthropomorphic here is like this particular species is mostly being identified in humans, and they are being transmitted mostly between human to human. So that's called anthropomorphic. So this is how cryptosporidium looks like. So the outer shell is the oocysts, it's called, and within this, it's the sporozoites. So when oocysts enter into the human intestine, then it releases the sporocytes, where the sporocytes goes and attaches to the intestine, and it does their work, and it leads to the infection. So what we know about this C. hominis? So what we know about this global epidemiology? So researchers have already identified that a particular subtype of this C. hominis, which is 1B810G2, is very dominant in developed countries such as Europe and US, where it is responsible for most of the human infections. It is one of the virulent subtypes identified to date. And in most of the waterborne outbreaks, this particular subtype has been commonly detected. So particularly in US, most of the waterborne outbreaks occurs in swimming pools, recreational water centers, and community water treatment plants. So through this, this particular subtype can infect humans and can transmit between communities. So in this study, what we did, we collected around 100 samples from around 18 to 20 different countries. And once we collected those samples, then we sequenced the genome of those samples. And then this is our bioinformatic workflow, where what we did is, like, once we got those whole genome sequences, that means in the form of sequencing reads. So we use those sequencing reads to map to the reference genome. And then we did some pre-processing and filtering to remove any low quality reads. And then after mapping to the reference genome, then we use that information to identify the SNPs, which is called a single nucleotide polymorphisms. For this, we use this very popular pipeline, which is a GATK pipeline, where you can use this for whole genome sequences to identify any SNPs or mutations in this whole genome sequences. So once we identified these SNPs in these samples, then we use these SNPs for different downstream analysis, which I'll be presenting further. So once we have this SNP, so our first idea was to look what is the structure within the population. That means whether we could see any clusters associated with any phenotypes within the population. So for this clustering approach, we first generated this principal component analysis. And very interestingly, we saw these two main different clusters. As you can see, this clade 1, which mostly involves the samples from low and middle income countries. And from clade 2, it mostly includes the samples from high income countries. That includes mostly from the Europe and from the North America. To confirm this clustering approach, we took another algorithm, which is a phylogenetic network-based clustering approach. And we also see these two divergent clusters. And we estimated that these clusters might have been differentiated or speciated between around 500 years ago. And further, we saw that in most of this clade 2, which is belonging to high income countries, so all these samples belong to this particular subtype, which I have just shown previously, which is a very dominant and virulent subtypes, mostly identified in high income countries or developed nations. While we didn't see this virulent subtype in this low and middle income countries, so that was very surprising for us. While except 1B810G2, while rest of the subtypes were highly dominant and present in this clade 1, which is belonging to low and middle income countries. Then our next question was, is there a genetic exchange happening between these two divergent clades? Because if there's a genetic exchange, or in other terms, we can say that recombination is happening between these two clades, that means they can produce very diverse or more virulent subtypes. So that would be very it's not a good sign in terms of controlling any kind of disease or understanding the pathogen evolution. So to analyze whether at any point of time there is a recombination or genetic exchange happen between these two clades. So what we did is like we compared the genomic sequences of these two clades, and we saw where a genomic region where there is a high sequence similarity between the genomes of two different clades. For example, this one. And this could happen when, for example, if a particular individual was infected by these isolates from two different clades. So once they enter inside the body of the human and they release their genomes, and that gives the platform for parasites to recombine and exchange their genetic material. So like this, we saw 3 genomic regions where a recombination or genetic exchange might have occurred between these two different divergent clades. So next, we wanted to investigate which genes are included in these genomic regions. And one more thing, whenever the recombination or genetic exchange happens between any two isolates, so that produces a novel SNPs. And when this novel SNP occurs, so that leads to higher variation within the genome. So that's why we see high variation in these peaks within the population. So that's within clade 2 and clade 1. And also when we compare these two different genomes from two different clades, so we also see there's a variation between the population. So this suggests that recombination is producing novel genotypes within these recombinant clades, which could be very virulent, and that's not a good sign for like when we would like to control any disease transmission. Then we saw which genes are present in these regions, and these genes, we found that these are the surface proteins which have the ability to go and attach to the surface membranes of intestines from any of the host. And also they are under positive selection. That means whatever the mutations that's generating within these genomes, it's very beneficial to the pathogen, and they can use for their own benefit to, for example, attaching to the intestines of the host. So we analyzed in the published paper, we analyzed all these major important genes, but for the example, I'll just show the story of one single gene here. So what we did, we generated the haplotype network of a gene, of a single gene here. So haplotype means like a stretch of DNA, which is shared, a stretch of DNA that contains the unique SNPs and that are shared between multiple samples. So for example, here there are total 6 haplotypes generated. That means you can say that there are 6 small clusters present here. So for example, haplotype 1 here is been shared by, for example, 10 samples, but here haplotype 3 and 4 are shared by maybe 1 or 2 samples. As previously, we have shown that at the whole genome level, this clade 1 and clade 2 are highly divergent. But at this gene, we show that a particular haplotype 2 is being shared by some isolates from clade 1, which is low and middle income countries, while it's shared between some samples from clade 1 and some samples from clade 2. So this suggests that when the transmission or when the recombination is happening between these two different clades, a virulent or dominant gene from low and middle income countries is being transmitted to, a dominant virulent genes from high income countries been transmitted into the genomes of low and middle income countries. So that means now, and this could be only possible when there is a high globalization have happened, where the isolates from two different countries or two different clades have come into close contact, and now they are recombining, and now they are transmitting into different geographical regions. So just to summarize this study, we have shown that within this C. hominis species, there is 2 subspecies which we have detected, which could be a result of subspeciation events. And since maybe they have been diverged around 500 years ago, but now there is a secondary contact happened, maybe due to the globalization or through other some processes where these 2 diverged lineages came into close contact and they got the opportunity to recombine with each other and produce new novel genotypes and spread into new geographical regions. And also we have identified a specific virulent genes that could be used for therapeutic development. So basically, we can use this kind of observations like whole genome genotyping studies for genomic surveillance or any outbreak investigation. For example, if you would like to, if an outbreak happened in any region, so with this approach, we can identify which particular clade is very dominant there, which particular clade is present there. So that will be very useful to identify a particular strain. And once we identify that particular strain, that can be used to stop the disease transmission. And the genes and the important genes what we identified here can be further used for therapeutic development. So and this is the title of the paper. If you are interested, so you can please have a look on this paper. So in the study 2, where we focused on another species, which is Cryptosporidium parva, it is a zoonotic species. So unlike Cryptosporidium hominids, so it is a zoonotic. That means it can be transmitted from animals to humans and humans to animals. So what we know about this Cryptosporidium parvum species from the literature. So from the literature, we know that a particular subtype, that is 2A and 2D, are mostly dominant in human infections that has been transmitted mostly from the animals into humans. And in animals such as the calf and lamb and sheep, these 2 subtypes are very dominant and very commonly identified. Also, the current literature based on this genotyping studies, they have shown that these subtypes are specific to geographical location and specific to host. That means a particular subtype, once identified in any geographical region, means identified in 2 different geographical regions, so their genetic content is very different. So for example, this is on the right hand side, there is a graph which is called isolation by distance. That means as the geographical distance increases, the genetic distance between these isolates also increases. So that means a particular subtype identified one geographical region is different from another subtype found in another geographical region or another host. So that's our current understanding before conducting the study. So for this study, we obtained 32 whole genome sequences mostly from different animals and humans. And we collected these samples from some countries from Europe, Egypt, and China. So again, we did the same, we undertook the same bioinformatic approaches. And our first question was to just look for any clustering or population-based structure within this population. So very interestingly, we found these 3 different clusters, which neither of them were associated with geographical region, neither of them associated with host. So that means whatever we saw to date in the literature was not agreeing with their observation. Obviously, the observation was only dependent on single gene or maybe the combination of genes, but here we are looking at the entire genome. So that's why we trust the clustering observed at the whole genome level. To confirm this clustering, again, we generated this network-based clustering approach, and we saw the similar 3 different dominant clusters here. So the lines or the loops between the clusters, which you can see here, that shows that there's a genetic relatedness present between the samples from two different clades. And this genetic relatedness only happened when there is a recombination or genetic exchange have taken place between these 2 samples from 2 different clusters. So again, we just tried to find how many or like the genomic regions where the recombination have taken place previously. And here also, like, it was surprising for us, unlike in Cryptosporidium hominis, where there was only 3 or 4 recombination events in the entire genome. But here we saw that around 73 recombination events were present in this Cryptosporidium parworm. And we just like, here, this is just a frequency of these events, and on the x-axis it's a year. So we just, for each of these events, we try to estimate when the recombination have occurred in the past. So the oldest recombination occurred around 16,000 years ago, but as you can see, the frequency is more high recently, and most of this suggests that most of the recombination have just started happening just 200 years ago. So obviously this could be the result of this increased globalization, as we can see that as international travel and global trade have recently been increased in last 1 or 2 centuries. And also, as we can see, this lot of recombination is happening in this zoonotic species, because it can happen between animals and humans. And compared to the humans, animals migrate most, because we know that there's an animal trade where there's export and import of meat and everything. So in this species, there's a lot of potential and lot of platform provided, where 2 samples from 2 completely isolated geographical regions can come into close contact and providing the platform to perform recombination or maybe undergo genetic exchange. So that's why, compared to the C. hominis, this species, C. parvum, can undergo more recombination. So again, we in the paper we have investigated those multiple recombination events, but here I would like to showcase just one example, the interesting case of recombination here, like how the disease has been transmitted from animals to humans and humans to animals back. So this is a network of a particular recombination region. As you can see that these 2 isolates from cluster 1 is completely isolated. It's getting segregated from its own cluster and coming closer to this cluster 3. And these 2 isolates or samples from cluster 2 is segregating from its own cluster and just coming closer to cluster 3. So that means there's a recombination has happened between cluster 3 and cluster 2 here, and producing this hybrid cluster, which now have the genetic material from cluster 3 and cluster 2. And same goes with this 2 isolates. So here was the same analysis we performed, and we found this genomic regions, highlighted this dashed black box. This is a genomic region where we see this high similarity between isolates from 2 different clusters, which gives us the more evidence that, okay, these are the genomic regions where the recombination have taken place. So again, we have taken that particular region and we performed this haplotype network analysis to see how these samples cluster together each other, clusters compared to the other haplotype clusters. As you can see, those same clusters, same samples here, for example, three samples from cluster 3, these 2 hybrid and these 2 hybrids now, they are sharing the same or similar haplotypes. The lines here between represents the number of SNPs differentiating between any of the 2 genome or any of the 2 samples. At the whole genome level, they are very divergent. They are isolated from each other, but now in this region where recombination have taken place, now they sit very close to each other and they share the same genetic material. Just to illustrate from which host these samples were isolated. So this is a schematic representation of how exactly in the past the situation has taken place. So for example, a cluster 3, a sample from cluster 3, which was collected from the calf, might have came into close contact from samples of cluster 1, and it might have transmitted the disease from animal to the human, and from human it might have came into contact with the samples from cluster 2, and it again transmitted back to the humans and to different animals here. And this might have occurred around 500 years ago, according to our estimation. So this shows that how when there's a close contact happens between animals and humans, so how a disease can be transmitted from a different host. So this is what happens, like, for example, if a particular subtype was initially not present in humans, but it was only observed in any of the animal, but due to some situation, for example, in the farm animals, like the farmers come into close contact with these farm animals, and if any farm animal have any infection, so there's a high chances that a particular infection from that farm animal can be transmitted back to the farmer. And for example, if the farmer has been traveling to another city or another town, and if he comes into contact with another animal or another human, then that gives that starts the chain of disease transmission, and that's how it spreads in a particular region. So to conclude and to summarize this study, where we have shown that the current literature, the hypothesis from the current literature is not perfect, and there is no clustering associated with geography, with geographical clusters, neither associated with host. Rather, there are some novel clusters, what we see now. The genetic exchange happening between these divergent clusters of C. parvum is fueling the evolution of this parasite. And as we have seen that as there is a contact between human and animal, so there is a high chances that a new disease can emerge into humans from animals. And the findings from this kind of approaches is very useful, particularly in studying how a new disease are emerging into the human population nowadays. And this kind of approach will be very useful in this One Health approach, which has been initially initiated in most of the countries now, where we try to understand, where we try not to focus only on humans, but we also try to focus on our surroundings, basically the animals, the environment, and everything, to understand how the disease is being transmitted. So this is the title of the paper where we published these findings. So if you're interested, you can have a look have a look at this paper. So this is the last study where I would like to showcase some other applications. So this is not a study here, but we have developed a tool here. So the basic idea of the tool is like, we know that these genetic variations occur at the genome level, but ultimately these genetic variations will affect at the protein level also. That was the main idea of developing this tool. Just to give you a brief background, in the recent years, there has been a lot of incoming of results, like published papers, which are mostly focused on genomics and genetics and genome-wide association studies. And in most of these studies, we see that a lot of polymorphism data has been generated. For example, in our case, as we have seen that we have generated a lot of SNP data from these two studies. And the main ultimate goal of this research is to like link this SNP or polymorphism data with the phenotypes. Like we try to understand like how this genetic variation or SNPs are ultimately being affected at the phenotypic level. And phenotypes, obviously, if you would like to understand the phenotypes, then we would like to first study the protein structure, because ultimately at the at any central dogma from genome to from DNA to RNA to proteins, so ultimate the result is the protein. So that's why we would like to we're interested to see what happens at the protein level. So when we wanted to conduct our study, so we saw that there's not much tools available that could allow us to integrate this genomic information on protein structure. There are few tools, but they can only deal one protein at a time, or a particular mutations or SNPs specific to that particular protein. but in our study, we are interested to integrate the genomic information from the entire population at the whole genome scale, and then study the effect of those genomic information at protein level. So for that, we decided to develop this tool. So then we developed this tool, which we named it as VIVID, which stands for Variant Interpretation and Visualization in Multidimensional Analysis. So the features of this tool is like it can showcase which mutation leads to residue interactions. So all these proteins composed of different residues, and if a mutation happens in one residue, then it will obviously ultimately affect its partner, which is present in that protein structure. So it tries to give the information which residue is being affected by that particular genomic variation. It also gives whether that particular mutation affects the stability of the protein structure, whether it affects its protein function. So for example, if there is a non-synonymous mutation happen, which completely changes the amino acid or the residue, so that might change the function of the protein itself. And it also gives whether the protein, it affects the protein evolution, whether a particular mutation is leading to its deleterious mutations, or it leads to the balancing selection or positive selection or negative selection. So this VIVID can interpret all this information from the genetic information, and it tells the users what happens at the protein level. And these are just a few examples which I'm just mentioning here, but there are many features available within this VIVID. For example, this is the graphical user interface of our tool. As you can this is a drop-down menu, and these are the various features where the users can click on these features, and it will just overlay all this information on your protein structure, and you can just visualize all this different information.
Hello, Dr. Saprin. You are muted.
Now, can you hear me?
Yeah, you are audible now.
Oh, okay, thank you. Yeah, so we use this VIVID for a new case study. And for this case study, we have focused on this another parasite, Plasmodium falciparum, which is responsible to cause malaria. So for this analysis, we focused on this vaccine candidate, erythrocyte binding antigen protein. So this protein can, once it enters the human body, it can goes and attaches to the protein of the human, and it leads to the infection. So that's why this is a very important vaccine candidate here. So here we show that, for example, here we show that how this vaccine candidate is having a different kind of evolutionary patterns in different regions. For example, we have these samples from Thailand and from Guinea. As you can see here, this is the region of the protein of this plasmodium, which goes and bind to the protein of human. So that's why the vaccine has been developed against this region here, so that it cannot bind to the human protein. So this circle is the very important region. So we see that this particular region is under positive selection, as you can see this more of red color here compared to the region in the Guinea population. So so in this study, we have shown that how this tool can be used to identify the differences, the evolutionary impact happening at different protein region. So that means if any vaccine has been developed against this protein or against this antigen, so it might be effective in Guinea population, but it might not be effective in Thailand population, because the evolution happening in Thailand is completely different from Guinea, and this might hinder the vaccine development, and maybe vaccine might not be effective in the Thailand population. So this is just one case study how VIVID can be used to understand the evolutionary genomics at the genome scale and at the protein scale. This is a link if you are interested. This is publicly available, and if you would like to try, please give it a shot. And this is the title of the publication where we published this tool here. So please have a look. So that's all from my side. So finally, I would like to acknowledge all the researchers and all the people who were involved in this work and supported throughout my duration. And also I'm thankful to this Cryptosporidium reference unit who have provided us the samples from all these different geographical locations. And without their support, maybe this study wouldn't have been possible. Yeah, thank you. I'm happy to take any questions.
Thank you so much, Swapnil sir. Swapnil sir, we have a question from Dr. Naveen Malik sir, and I now invite him to come and ask you the question.
Yeah, sure.
Hello, can you hear me, sir?
Yes.
So, very nice talk, sir, Swapnil sir. So I'm Dr. Naveen Malik, and I have a little query. So basically, you talked about different subspecies of Cryptosporidium in your evolutionary genomic studies. So my question is, obviously, you're working in, you're talking in the USA context mainly. So can you basically tell me some subspecies or a group of subspecies which are more important in terms of Indian context? If you could find out some subspecies of Cryptosporidium which are more important in terms of Indian context.
So you're saying that if there are any subspecies been detected in India?
Population. Yes, yes.
Yeah. So I think, to my knowledge, there were very few studies that has been conducted in relation to the Indian population. There was, I think, only 1 or 2 samples. I think in my data set there are one or two samples that's been that we used in our study. Let me just give it. But nothing was dominant in this population. So for example, here in this low and middle income country, I think maybe we have 1 or 2 populations from India, but that's definitely not belonging to this 1B, 8, and G2. So we can't say at this moment whether the particular infection that's found in India is really dominant or highly virulent. So I think we still lack the data from the Indian population. Yeah.
Okay, fine. Thank you, Dr. Swapnil. I have another question from you. So basically, you have worked on evolutionary genomics and find out different subspecies of Cryptosporidium. So my question is that have you basically? I'm trying to know whether it is possible to find out some association mapping kind of approach for these kinds of studies. Because I don't know, because these kinds of studies are important only in those scenarios where your trait is quantitative. So I don't know whether diarrhea is a quantitative trait. So different variants or different severity of diarrhea is present or not. And in that case also, if we have Cryptosporidium subspecies, whether these subspecies are diverse enough to take as a different species. So basically, they can be utilized for association mapping. So my question is, can these informations be utilized for association mapping studies where we can find out the causal gene, a causal SNP, which is mainly associated with their disease severity in case of diarrhea? So I hope you understand my question.
Yes, I think I understand. Yeah, and that's really interesting question. Yes, so definitely, for example, the most of the researchers have focused on this 1B, 8, and G2 subtype here. So that's very kind of it's a popular or it's a celebrity in this cryptosporidium subtypes, I can say here. So yes, there's a severity associated, but no study has been conducted yet to associate this any of these subtypes with the severity. So mostly for the severity, for example, we try to investigate through the stool samples. So in the stool sample, for example, we see how many like, for example, the sporocytes, or how many the oocytes, or like oocytes. What I mean here is this is oocytes. We try to measure in the stool samples what is the proportion or what are the numbers of these oocytes. So if it has more oocytes producing in the stool samples within a particular window, say for example within 24 hours or 48 hours. So that's how currently they are measuring the severity of the this disease. But associating with them any specific subtype or species is still not been conducted. Research on this one is still going on.
Mainly with the specific SNPs, if we found them in different subspecies. So it can be that studies can be taken if, but that main question is that species should be diverse enough, and that rate should be quantitative. If it can bring this area can be explored.
Yes, for this SNP thing, so currently mostly people are focusing on only single gene, which is GP60 gene. So you can have a look. So that's mostly focused on, but sometimes that might not differentiate between different strains, GP60. So that's why like we are trying to propose that rather than focusing on maybe on the single gene, maybe it's better to focus on whole genome sequences. So maybe this kind of study may be conducting the qualitative, association between the severity of disease maybe further would be useful. But currently, yes, still there's a lot of limitations in this field.
Okay, thank you, Dr. Swapnil. Thanks.
Thank you so much, Swapnil sir. It was indeed a pleasure hearing you. And on behalf of the organizers here, I would like to thank you for providing us a very valuable insight on a new perspective towards research in genomics. Thank you so much, sir, for joining us. I know it might be midnight over there, but thank you so much, sir. Thank you.
Yeah, thank you very much, and thank you for having me. Yeah, have a wonderful day.
You too, sir. Thank you. Moving ahead with our session, our second speaker for the day is Dr. Amandeep Singh Pannu. And sir has joined us. Morning, sir. Sir
Morning.
has joined us from Queensland University of Technology, Queensland, Australia. Sir is a graduate in physics and nanotechnology with a bachelor's in maths, physics, and chemistry. He was awarded his PhD in material science with a specialization in fabrication of optoelectronic devices from the Queensland University of Technology in August 2021. His research experience spans over 6 years, including his PhD candidature. His PhD work was focused on the development of advanced materials for optoelectronic device and energy storage. In his research, he successfully developed new materials and demonstrated their applications in sensors, organic flexible light emitting diodes, solar cells, lithium-ion batteries. Currently, Sir is working as a visiting fellow at Queensland University of Technology and a research fellow at CSU. So with these few introductory words, Sir, we take the honor and privilege of welcoming you here. Thank you so much for joining us.
Thank you so much for your kind words. Before moving forward, I would like to share my screen.
Yes, sir.
Yeah, sure. So can you please confirm that you are able to see my screen?
Yes, sir.
Okay, thank you. Okay. First of all, I would like to thank thanks the organizer, especially Dr. Pushpinder, Professor Pushpinder.
Hi, Aman. Hi, how are you?
I'm good.
Really,
Good.
thank you so much for joining, and taking time to really talk in this particular conference. I'm really, thankful.
No worries, no
Dr.
worries. This was
Malik
my
will,
pleasure.
Dr. Malik will speak more about your, about you. And because right now I'm busy with some academic work.
No
And
worries, no
yeah,
problem.
yeah.
No
Thank you so
problem, no
much.
problem.
Okay.
No problem. Okay,
Okay.
okay. So without wasting, thank you everyone for joining. Without wasting my time, I would like to present my work because we are, as I know, we are running a bit late. So my work is basically based on synthesis of advanced material and its application as well. So we were not limited to just manufacture the material, but we had a vision that we need to demonstrate its application as well. So we design the material, we control its property, and finally demonstrate its application as well as a whole. So so I will be discussing here. So I have demonstrated this material which we have made from waste material in light emitting diodes, in solar cells, in lithium ion batteries as well. So, but today I will be limiting my discussion to three main, which we have published in recent times. One is that sensing, which we have with carbon dots. Next is we have developed a two-dimensional carbon dot emitter for light emitting diodes. And third is that we enhance the overall stability of perovskite-based light emitting diodes. So we'll discuss all these in detail. So starting, when I started my work, lots of people asked me that while selecting a waste material, why did you try just waste humans hair stands? So what happened is that we were when I started my work in sustainability and as a material scientist, so we were looking into a waste which can be easily handled, and its logistic is very important, because if you have a waste which is biodegrading and it is wet and it is leaking all the time, handling of this waste is very difficult. So we were looking into the waste which can be easily handled, and which ends up in a landfills. So we started from human hair. Now we are working with wool, waste clothes, and lots of other exciting things. So which I will discuss at the end. So in this study, what happened is that we used waste small strands, and we try to make carbon dots by using two heating methods. One is hot air oven, and another is microwave. We had a commercial microwave apparatus in our lab. So once we manufactured, we found that the final material, if you can see at the very right end, you can see this material, once it is exposed to UV light, it starts emitting in a visible region, a light blue color. So we had this two materials with quantity yield 17 and 38. It depends upon the yield, depends upon how much number of photons it is observing and how much it is emitting. So it was observing in UV region, but it was emitting in a visible region. The below figure B, C, D, E is the transmission electron microscope, where we were trying to visualize this nanostructure. What is the distance between two atomic planes? If you can see at a high resolution image of E, if this is so you can see that the distance between two planes of carbon is 0.23 nanometer. Okay, so this was the basically we're investigating its properties. So what was the motivation behind this? So the motivation was that when we were synthesizing this carbon dots, basically we were trying to introduce other nitrogen containing precursor into the material. So why we were doing that? So because we were trying to generate a nitrogen containing moiety at its surface of a carbon dot. So there was two possibilities. So one is that you have a bond of nitrogen with the carbon. So you have a CN. We call it as a pyridine nitrogen. If you can see at the figure C, pyridine nitrogen. But if nitrogen combines with the oxygen embedded into the carbon as a host, so we call it as a pyridine N oxide. So what we were interested was to create pyridine N oxide more as compared to just pyridine and CN. So we were much more interested to create this NO moiety, and you can see that in MCDs, the carbon dots which were manufactured using microreactor microwave reactor, we were able to create a lot of NO moiety as compared to NH2 and pyridine N. Why we want to create NO? That was the next question. So what happened is that this we found that through computational analysis, we found that this NO has a particular dipole moment. So when you create this NO moiety over a carbon dots, it has a strong dipole moment, and we have in figure A shows the different hybridization of charge moving around the structure. And what happens? We were trying to sense chloroform, and we know, as we know, that chloroform also has some dipole moment. So what happens when you bring lots of other molecule and chloroform is presence in this impurities? What happens? There is a strong interaction between this NO structure and chloroform, which helps to increase the fluorescence of the material. So if you can see in this figure, what we happened is that we have to demonstrate that in a normal, in a potable water also. So as you are aware that normally most of the water supplying corporation, we call it as a corporation over here. So when government supplies water into your home, they disinfect with a chloroform. And when the disinfection process of adding chloroform to the water creates, chlorine, when the government adds chlorine to the water in a water for disinfectant, this chloroform is a byproduct and it is carcinogenic in nature. So we were tasked to detect into potable water, but there were other impurities as well present. So we demonstrated in studies in presence of other moieties, because no other molecule had a strong dipole-dipole interaction except this chloroform molecule with our carbon dots. We were able to demonstrate that the graph on the left-hand side shows that as you increase the chloroform's concentration, the fluorescence of the material increases and it saturates at one point near 100 ppb. So we demonstrated as compared to other studies which were present at that time, we were able to detect lowest possible concentration of 3 parts per billion, 3 ppb, and we have published this work. Okay, moving on. So I was quite confident. So as a team, we were able to demonstrate that we can control the structure of carbon, what we were manufacturing from waste, and we can precisely control by variation of temperature, addition of precursor into it. So the next demonstration, basically I was working in a device lab where we were manufacturing light emitting diodes, transistors, and solar cells. So my PhD was based on demonstrating a device as well. But when I found that when I joined the institute, I found that people were talking about cheap, printable, flexible devices, but they weren't cheap at all, because the material which was going into these devices, the active material, was more expensive than gold. So my research was basically to demonstrate a very cheap material so that we can call them a cheap, but we can actually sell them as a cheap device as well, because otherwise it's a very expensive device to make. People are just telling that they were cheap, they were not so. Okay, so the next step was to demonstrate, since the carbon was emitting emission when excited. So the next step was to control its emission properties, not to control its sensing property, but its optical emission. So what happens when people manufacture these carbon dots? 99% of these carbon dots are like in figure A. So when you excite this carbon dot, they start emitting, and when you change the excitation, their emission also changes. So this is called excitation dependent emission. So which is very wrong, because if you excite the carbon dots as one wavelength, its emission is staggered over various wavelengths, which is not good for a device. We need to have a at whatever wavelength you excite, its emission has to be at one point. So we were trying to have excitation independent emission. So what we did is, so the very first thing I optimized is the temperature. What was the best temperature to get a very defined crystalline, a single carbon dot? So we found that it was 240 degree. Then I applied its, then I controlled its surface properties with ligand to increase the summation, and we got a best material at 240 degree with a photoluminescence quantum yield of 87%, which was almost near to highest reported at that time. So the next thing was that I increased the concentration of ligands around this carbon dots so that its emission is independent of excitation. If you can see the figure 5, so we use laser at 330, 340, 350, 360, and 370 nanometer, but the carbon dots always emitted at same wavelength. Only the intensity was different, but it tends to emit at the same wavelength. So which was very important. So you need to control the properties depending upon what application you have. So we were trying to control its optical property. The next step was what happened normally. When you want to use such type of material on a thin film on a solid state. So these carbon dots, people manufacture them and they emit very different, vivid, very beautiful colors in solution in water or other solvents. But on solid state, their emission is quenched. This is a basic problem, quenching of nanoparticles when in a solid state. If you can see figure E, these are the carbon dots. Without any other modification. If you can see, this emission is very, very less in intensity. But if you can see the figure below, where we have made a carbon dot islands, you can see we were able to preserve its emission. So what happened is that we try to self-assemble these carbon dots and make small islands, and these small islands were again protected by ligand. So this happens all on a spin coater. We add the carbon dots to the ligand at a particular ratio in a two-step process. While spinning, we were able to form a very thin film. You can see a scanning electron microscope at we take at a very big scale to demonstrate that it has a long range order. And in figure C, you can see an AFM image which demonstrates the exact. You can see a very beautiful image of all the carbon islands. And if you add also measure the mechanical properties with the tip, as you can see that all the islands were protected by, you can see variation of forces around the perimeter of each carbon island. So the figure F shows how. So see, this is a thin film, very small emission in dark blue color, and this is the emission from carbon dot islands, which we demonstrated there. So we were able to demonstrate very first time that just with using carbon dots only, demonstrating to forming a self-assembled structure, their emission can be preserved. So finally, we were able to demonstrate a device. So these carbon dots were supposed to excite by electric current, not by UV light. So so my lab is specialized in making such thin film devices. So we used a glass coated with ITO, and then we used a whole transporting layer, P.PSS. And over that, we are using again a PVK and CD. So these are the whole transporting layer. And over that, this TBPI is an electron transporting layer, and aluminum is again an electron injecting layer. So basically what is happening, so you attach the positive terminal here and negative terminal to the other electrode, and you inject opposite currents. So positive means holes and negative means electron. So when these holes came through the P.PSS layer, goes through PBK, and rest came into the carbon island, and then electron came from above layer, and they both meet there in carbon island and produces a light. You can see in figure B. So once you switch on this light emitting diodes, you can see the emission from this device. Further moving on, we want to probe its structure also. So what happened is that we used a focus ion beam to cut a very small cross section of this device and analyze it further. So this was the very first demonstration, and we were able to publish all this work in advanced material, which has around a impact factor which was best in my field about, it has an impact factor of 30 around. So what we did is that we used a focused iron beam and we cut a very small cross section and picked it with a tweeze, a robotic arm inside the device. And then we load the same into a transmission electron microscope. If you can see this figure F, you can see we were able to visualize each layer. So this carbon dots is around 40 nanometer, and you can see the crystallinity of all this carbon dots island formed in a devices. So this was the first ever report to demonstrate the internal structure of material, the device. Moving on, also we manufactured the flexible device as well, not only the rigid. So we used the PET substrate, and we manufactured all the layers to the top, as shown in figure before the last slides, and we were able to demonstrate a flexible device. So again, this was the very first report to demonstrate a device. You can see clear in figure C, you can see the PL and electroluminescence. PL is photoluminescence of device. Electroluminescence means when it is switched on by an electricity. And the figure F shows its entrant structure, which was used by, we used helium ion microscope, because if you put up a very flexible device into a TM, it will burn whole device. So we need a non-destructive technique, and helium ion microscope was one of it. So we used HM to analyze the internal structure, and we can clearly visualize the carbon dot island inside the device. So this work was covered by lots of websites and newspaper. The Brisbane Times, where I was situated, PUT is situated, also covered as SBS Australia, ABC, Australian Broadcasting Corporation. Everybody covered our news, because it was the very first demonstration of light emitting diode, where the emission layer was made from waste human hairs. Moving on, I will take a few more minutes to wind up my talk. The next work were demonstrated. So there were lots of interest on perovskite based material, but one major problem with perovskite material is that it is not very, it is prone to it is not stable. Actually, it is prone to moisture. Basically, it is a salt. So when salt is exposed to air, it observes the moisture and disintegrates. So this is the problem of perovskites. Simple problem. So you need to protect it from the moisture, or you need, so people are working on various aspects, how to protect, how to make a robust structure. Anyhow, so so one of my colleagues were making this perovskite, very beautiful perovskite crystals, cesium lead bromide. But the problem was that once you make an light emitting diode out of it, over the time it disintegrates. So it will work very fine on very first day, and after second day's emission is gone. So we were investigating how to protect this layer. So this is where our carbon dots came. So we made a carbon dots with a layer which was hydrophobic. This means it repels water and it doesn't interferes with the working, but at the same time it helps to move the charge across the layers. So what we did, is we mixed the perovskite, we deposit the perovskite and deposited a very fine layer of carbon dots, and demonstrated that, if you can see, so this is the dark blue color is the perovskite with carbon dots, and the light blue is the perovskite, just only the perovskite. You can see these two structures. So we were optimizing these two structures, and we found that when it is exposed to moisture, it remains again stabilized. The surface states remain stabilized. We demonstrated it there. You can see that. So this is XRD image of this, and we were demonstrating where are the bromide state and where are the lead states are there. Then we also measured its emission. So how fast is the emission? did carbon dots coating affects this emission, because perovskite emission is the time delays in few nanoseconds. So, and then we actually measured the valence and conduction band of these two layers. And if you can see, so this is again same device. We use the same structure: glass, P.PS layer, composite, perovskite layer. This material had a better operational stability. So this is so once you have this composite material, the offset between charge injection, offset means holes is going into this, and minus 5.1 and 5.3. So this offset is reduced. And so the whole of the device, the composite, so if you can see at the figure C, you can see the actual pixel of this device. And then we compared the pristine, if you can see current density, also figure B, pristine device, which doesn't have carbon dots, and a device which has carbon dots. We demonstrated that, in fact, because of the reduction of this offset, actually carbon dot composite with perovskite was helping to reduce this offset and helps in injection of holes better, so that the current density of the final device was always better at all the voltage where it was operated, from 4 volt to 12 volt, and all the ranges, its current density was much better compared to the pristine devices. Apart from that, we also demonstrated, if you can see, this is the lifetime analysis. The normal pristine devices decay less than 50, like in first 20 hours also. But with carbon dots, although there was still we weren't able to produce the robust OS because the coverage was not very good in very first studies. But we were able to, so you can see that large increase till it was 50% reduction, and it has already operated for more than 200 hours. So from just 10 to 20 hours to 200 hours, we have demonstrated the devices were working better. And at the end, so not only stopping here, so we have also demonstrated where we can control these carbon or nanostructures and we can form various structures. In figure A, we have shown that we can form pillars. This is a SEM which shows a long range order, and then there is an AFM at the bottom which shows actually it's a pillar structure, it's not a sphere, and we are trying to use this as a charge injection layer to improve the performance of devices. Apart from that, we have my recent project where we have recently published a paper. We have demonstrated, because I'm a material scientist, we were working to make from waste wools, waste clothes, we were able to demonstrate manufacturing graphite on demand manufacturing graphite, which has lots of companies have shown a large interest, and soon we are going to start I am going to start my own lab on this technology. We are going to commercialize this technology further. And thank you all for patiently listening to my presentation. So first of all, I do acknowledge my mother university, Queensland's University of Technology, then funding from Center of Waste Free World, QUT, from Charlestown University support, and from CARF, which helps to analyze all my, so it helped me to provide all the machine instrumentation and support. Thank you all. Yeah, I'm open for any question from here. Thank you, everyone. So
Thank you so much, sir. Sir, we have question from Dr. Naveen Malik, sir, and he's coming forward to ask you the question. Naveen, sir, please.
Good morning, Amandeep sir.
Good morning. Good morning. Yes.
So it was a very nice talk delivered by you, and very enlightening talk, and especially on the area of nanotechnology. We have used basically synthesize various kinds of nanoparticles from bio waste and other sources as well. So it was really a very nice talk, and we are very happy. I think all the audience have benefited from your talk, and I have a few questions basically. So in your slide, you talked about bio waste utilized for high performance carbon emitters. So can I basically know what kinds of bio waste was used there?
Okay. Yeah, bio-waste, right. So basically, we have used small human hair strands to make carbon dots, but now we have extended our study to make it from waste wool. So we are in contact with lots of companies here. We have a huge wool industry in Queensland. So we are working with them. And apart from that, there is a company called Fast Fashion. So they have discarded clothes where one of my friend is separating all the different kind of material from the clothes. And then they are supplying us which of the material has the highest carbon content. Basically, we need a carbon content from starting to convert it into our nanostructures. So we have diversified our precursor material. We started from human hair. Now we are going into wool, waste woolen clothes as well, and waste other clothes as well.
Okay, very great work by you, sir. And another question, basically, we have in this current scenario, basically, if we talk about in Indian context, so plastic is a very, you can say, area of concern. So can we use these plastics as well for generation of these carbon emitters or nanoparticles so that can be utilized further?
So we haven't investigated. But we definitely. I think other people might have tried. But anything with carbon can be converted, but it depends upon whether we can convert it into emission material or some other structure. So it is very important as well. So sometimes you aren't able to convert it into emitter, but same material you can convert into, you can graphitize it more. Like under a controlled reactor, you can make a graphite out of it or a graphene out of it. So it depends upon the type of material. Maybe all plastic we haven't looked into, but definitely we are going to look in a future, because my current project, we are making a lab-based reactor, which we are going to commercialize as well, where we are getting very high temperatures. What is the problem is that normally in any given lab, you don't have a reactor which can go beyond 1200 degrees. You have tube furnaces which can go at 1200 under a atmospheric condition under different gaseous situation. But if you want to go beyond 1200, even 1800 or 2000 degrees, so and basically, so there is no lab reactor. So only few labs in the world which are called high temperature synthesis labs have that. So we are into that. We are trying to manufacture a reactor which uses very less energy. It is called wave reactor, short waves. We created a small waves, and we are trying to manufacture this novel reactor so that we can reach high temperature, which is needed to convert such type of material.
Okay. Thank you, Dr. Amandeep. And your talk was very informative. And basically, I want to tell the audience as well that Dr. Pannu have expertise in various fields like nanotechnology, physics, and chemistry. And these kinds of interdisciplinary research can be very important. Specifically, his expertise can be of great importance in developing or finding sustainable solutions for various biological and engineering problems. And this greatly resonates with our current theme of our conference. So it was really a very good talk by Dr. Amandeep Singh Pannu, and we wish him all the best. And we thank you as well for accepting our invitation to deliver this talk here, keynote address. And I'm very thankful to Dr. Pushpendra Sharma. He is very personal as well as professional links with Dr. Pannu in various capacities. And thank you, Dr. Amandeep. And looking forward for inviting you, or basically you can visit the university in person in future as well, we'll be more than happy to.
Absolutely,
be here.
absolutely. We are going to start our own lab and we are getting investment. So I'm going to start up a company as well. So we are basically going to develop one of the company, and definitely we will look forward to visit India as well. Yeah, the
I
Amit
must,
is on my card.
Amandeep, I must say that, your hunger for science will never end. I think I have seen you in the university actually. It's really wonderful
thank
to
you.
see your work. It was really, I think, a transformation in a technology that you're using. I couldn't
Thank you
really
so
stop
much.
myself to appear, even I was busy. But yeah, I want to really, it was amazing work. I think it's a wonderful work. Great.
Thank you so much.
I wish
Appreciate your support.
Yeah, I wish you all the best for your, endeavor that you're doing. Definitely, you will come up with great solutions for sustainability. Definitely.
Yeah.
Thank you so
Thank
much.
you.
Thank
No
you.
worries, you are
And,
welcome. And at the last, I want to apologize that you have heard my daughter voices because I am presenting from my home. It's like 5 o'clock, and sorry for that.
No, it's all right. Thank you.
Okay,
Thank you.
bye. Bye.
Thank you, Dr. Panu. Thank you. Thanks a lot.
Welcome.
Thank you very much, sir.
Other than the registered participants, if anyone wants to go for the opening ceremony of the Sangathan, he or she may leave. Continuing further with our sessions, now we are beginning with our next oral presentation session. The session will be chaired by Dr. Sudarshan Singh Lakhawat and co-chaired by Dr. Naveen Malik sir. Dr. Sudarshan Singh Lakhawat is CSIR NET qualified and holds MSc Biotechnology and PhD Biotechnology degree. Sir is currently working as Assistant Professor in Bioprocess Engineering at Amity Institute of Biotech, Amity University, Rajasthan. He is having more than 18 years of teaching and research experience. His area of research is in kojic acid production process, bioethanol production, and I request the audience to maintain silence as well. Bioplastic production and plastic degradation process. He is skilled in using lab and pilot scale fermenter. He is having more than 30 research and review articles in national and international journal of repute. Sir, we welcome you as the session chair for this particular session. Dr. Naveen Malik is currently working as an assistant professor at Amity Institute of Biotech, Amity University, Rajasthan. Previously, he had worked as postdoctoral fellow at National Institute of Plant Genome Research, New Delhi, from 2017 to 2021. He has earned his PhD degree from NIPGR New Delhi in 2017. Sir has also been awarded with a startup research grant of rupees 32 lakhs by DST Serve in 2022. He has more than 14 years of teaching and research experience. His research work is focused on genetic dissection of yield and architectural traits in crops, especially rice, chickpea, employing reverse genetics and association mapping approach. His research interests also include molecular characterization of phosphate deficiency in pearl millet, especially through omics-based studies. Sir has published 25 research papers and review papers with a cumulative impact factor of more than 140 and an H index of 12. Moreover, he is currently guiding PhD students and few undergraduate and postgraduate students for their thesis and doctoral projects. So with these few words, I hand over the session to Naveen Malik sir and Sudarshan sir. Welcome, sir. Thank you. And I request audience again to maintain silence.
So, very good morning to all of you and all the dignitaries which are attending these sessions, the previous speakers, different faculty members, organizing chairs, participants, as well as student volunteers. So I welcome you here and a very warm welcome from you, from my side and from side of Dr. Sudarshan Singh Lakhawat. So we have basically 10 speakers in this session, and one by one I call their name. So only one person is attending this conference in person, others will be available on online mode. So the first speaker is Mr. Room6, and I think he's sitting here, so he can come here and deliver his talk. So welcome, Mr. Dhruv.
Okay, so see, the guidelines are that you will be given 5 minutes for the presentation and 2 minutes will be for the viva voce or question session. Okay, so you can follow that guideline. Now, regarding the online participants, once the presentation is over and the session is over, then you need to unshare the screens also.
So, hello. So, everybody here might be knowing or might be having some basic knowledge about fruits and vegetables. They have great demand in national and international markets. But on the other hand, they are perishable in nature as they have very short shelf life. Good morning, I am Dhruv Desai, and I would like to share some information regarding my work, which is on the topic edible coating for fruits and vegetables, which is basically a review. So, edible coatings can be a great solution to the global problem of perishable fruits and vegetables, as they act as barrier. It's okay, you can continue. I think.
IT support? IT support? Excuse me?
Hello. So why review paper, or what is the need of a review paper? So basically, there is ample of amount work done already in this field. So there was a need to keep the basic knowledge in the one single paper. So the main objective of this review paper was to provide a brief overview about the recent advancements that has been done in the coding preparations. So, and secondly, the second main goal or aim was to understand the importance of edible coatings, as it act as a barrier for gases and microorganisms. And secondly, most of the edible coatings are biodegradable in nature, and hence it will also contribute to sustainability. So now, the recent advancements. So the recent advances that I was talking about are, firstly, development of smart and intelligent films. Then secondly, special type of polymer based materials were used which will protect against the physical damage. Third, coatings developed from bio-nanocomposites, which will help or result in a stability of biomolecules. Fourth, nanotechnology. Nanotechnology-based smart and intelligent films resulted in the extension of shelf life much better than in comparison to the coatings, in comparison to traditional methods. And fifth, metal oxide nanoparticles, which included silicon dioxide, magnesium oxide, zinc oxide, and iron, etc., which can act as food additives and antimicrobials. Now, all these recent advancements has been reviewed in one single paper. So the methods that was discussed earlier, that 4 methods and these recent advances, that all it has been mentioned in one single paper. Now, this review paper will now help the readers or researchers to understand the importance of edible coatings and what type of research has already been done. And this will help, and this review paper will help to find the research gap or research problem, and hence finding the solution, which will help to the society. Now, acknowledgements. I would sincerely like to thank Dr. Arpit Srivastava, sir, for his expert guidance throughout this journey. Additionally, I would also like to thank MIT University for giving me this opportunity to present my work at this stage. Thank you.
I have one question that would it be economic to use such type of coatings because it can increase the cost of the food overall?
Yes, sir. But actually, fruits and vegetables have less shelf life as they are perishable. So if we are using this type of coatings, then the shelf life will be extended. So yes, that it will increase the cost of fruits and vegetables, but then we are getting the shelf life, extended shelf life. And it also can provide a substitute to the films or coatings that are prepared from biosynthetic materials like plastics and all. So it's much better substitute.
Status. I mean, are any type of the coatings being used right now?
Yes, actually, research has been done like on herbal technology based coatings. Then some are like polysaccharide based like starches. Then proteins and all, which I mean, smart and intelligent films also has been developed in this. Like for example, like chitosan, they are using some plasticizers like glycerol and all. Then anthocyanins are being used for intelligent packaging, which will then work as a detectors, like the color of the edible coatings will be changed. So after some time, like if it is getting spoiled or fruits inside.
What is the code of action? Like how it prevents the
Actually, it acts as a barrier. It acts as a barrier for against the gases and microorganisms. So hence this will increase the shelf life.
Okay, thank you, Dhruv, for your nice presentation. So we have in the queue next speaker is Mr. Alok Kumar Singh. We are sitting here. Please come. Over to you, Alok.
It's should be repeating. That is why. It's okay. ठीक है चलो. Good afternoon to everyone. Myself, Alok Singh from BSc Biotech third semester. I am here to present my work on bibliometric analysis of food health risk associated with chemical in foods. Let's start. Certain chemical in the environment can find their way into the food chain and pose human risk to human health. Several categories of these chemicals are present in environment like heavy metals, pesticides, persistent organic pollutants, industries, chemical and byproducts. These chemicals persist for longer time enter the food chain. These chemicals are highly toxic and produce harmful effect to the environment and human beings. Chemical in food can serve various purposes such as extending shelf life, enhancing flavor, improving texture. However, some chemical used in production can pose health risks and toxicity problems if consumed high amount and prolonged periods. These chemicals also affect the nervous system, kidney, and other reproductive issue, neurological disorder, hormonal imbalance, cancer, and other health concerns. In this study, bibliometric analysis was utilized for the data extractions as bibliometric is a method that combines mathematics, statistics, and related discipline to analyze documents and uncover relationship among them. Article details such as affiliations, countries, authors, journals, and research area and keywords were extracted from Scopus database. In our analysis, we use bibliometric analysis by RStudio and WASViewer for graphic visualization and mapping.
Sorry to interrupt you.
Yes,
Can you basically tell me the topic of your talk? In what context you are talking about? Topic.
Chemical in food. A bibliometric analysis of chemical in food. Okay. This graph shown here represent the global impact of publication in the field of chemical in food and health concerns between 2022 and 2024. The most cited paper authorized by Barbosa et al. In 2022 has received 518 citations. This indicate major influence of their work, which was published in the Journal of Science of the Total Environment. This graph shows the country collaboration present here, illustrate the geographical distribution of the publication. The map show that China, here is the China. The map shows that China is a significant player in this field. Field is as evidenced by the dark blue color. Dark blue color indicate the highest number of collaborations with the various countries. According to these threshold values, the total number of the cited author from this study, the data shared meta-analysis 753, this revealed that 102 author have not been cited. This graph represents the author network analysis for the publication related to the chemical in food. It visually map the connection and collaboration between the various author who have contributed in this field. This graph shows the relationship between the journal of the field of chemical in food research. This color represent the different cluster of the journal that are closely related in the term of the topic they cover in the citation pattern. The size of the node proportional to the number of the cited paper. Here are the prominent keyword we used. The main keyword is human, which is closely placed in the fish, nickel, and soil, showing the effect of pollutants. The impact of the research in this field is evident from the increasing number of the highly cited paper, particularly those published in the top tier journals such as the science of the total environment and chemosphere. The anticipated that research on chemical in food and health risk will continue to grow, particularly in area addressing the interesting intersection of the climate change and food system and health outcomes. The prominent keyword analyzing emphasizing the chemical in food reference human, the food and water contaminated through the chemical and heavy metals in the environment, which poses risks to the humans. Acknowledgement. I would thank to Dr. Shweta Kulshrestha ma'am and Amity Institute of Biotechnology and my colleague Priya for her support. Thank you. These are the references.
What was the source of your data cycle? What was the source we are used for collecting the entire data?
So first we use Excel. First we put data in Excel sheet, then program in R Studio. Then we use WasWeaver to make the graphs and maps.
Which search engine you have used for searching all these type of papers and you have used some kinds of search engines from
Google Scholar and PubMed.
PubMed.
Yes, sir.
With the keywords, what are the keywords you used?
Mainly, sir, we focus on human, fish, heavy metals.
And how do you decide that these are the top tier domains? These are that these two chemosphere and science of
So,
total environment is top tier.
sir, more paper are published in this journal only. There are 518 paper are published in this journal. Barbosa et al.
My concern is basically you are taking data from Google.
Yes, sir.
Google Scholar.
Yes, sir.
So in that context, basically you are putting some keywords there. So these keywords may not be sufficient enough. So because you are having hundreds of chemicals with food, so you're not using those keywords. You cannot use all of those keywords for searching the database. So how are you sure that only these are the papers which are available there and not others?
Sorry, sir.
Because you're saying that it's a bibliography data, so I think you should have covered all the things here. But I'm skeptical, like you haven't used all the keywords here, so that they miss.
I used few keywords, major keywords are like human, fish.
You have talked about chemicals in food.
Yes,
You
sir.
haven't used chemicals. There are hundreds of chemicals there. Or you use those chemicals that use food adulteration. Like, so is this the actual data, bibliography data which is available, or much more data is available which you haven't covered?
So I work on. Thank you, sir.
Okay, thank you, Alok. So now we have next speaker with us. So that is, she is RV Chen. So RV, are you ready with your presentation?
Yes, sir.
So can you share your screen?
Sure, sir.
Are you having any issue?
Yeah, no, sir, just a moment. I'm just sharing it.
Arubi, have you shared the PPT with our volunteers? Yeah, it is coming. Is it the flame test one?
Yeah, ma'am.
Okay. Okay, so please make it full screen. It is here now.
Ma'am, is the full screen visible to you?
It's not full screen. Can you make it full screen?
Yeah, I did. It's processing, I guess. One second.
Seems like multiple PPTs are open at your end.
Ma'am, I will just share the PPT with the team.
PPT is here. Okay, you do one thing. You continue with your presentation so that we can save time. Even though it is not full screen, continue.
Okay. So good afternoon, everyone. My name is Orvi. I'm a graduate student from Amity University, Rajasthan. So you all might be well aware about the spread of coronavirus and the widespread use of hand sanitizer. So I would like to present my today's presentation on the production of bioethanol and its application as a hand sanitizer. So for this, I use three substrates, honey, sugarcane juice, and palm jaggery for the bioethanol production to be used in the hand sanitizer formulation. And each substrate were fermented by fermentation to convert sugars to ethanol using the brewer's yeast S. cerevisiae. And bioethanol from these sources was possible by careful use, safety measures, and verification was crucial at every step. That was did. I will explain that further. The formulation of hand sanitizer was done by the WHO World Health Organization Guidelines Protocol. Talking about the materials and methods. Talking about the materials and methods, these were the substrates used and the chemical used were for different methods. These were sugar estimation was done by phenol sulfuric acid method. In this, the spectrophotometer was used to check the OD at 490 nanometer. Ethanol estimation was done by potassium dichromate method, and fermentation was done for each of the substrate by using the inoculum S. cerevisiae by adding them in these figures that you can see. Distillation was done for each of the substrate and a minimal amount of ethanol was produced. And this is the hand sanitizer formulation that was done using the WHO guidelines. The materials used were ethanol, glycerol, distilled water, and hydrogen peroxide. And it was shaken vigorously and kept quarantined for a few days so that the hand sanitizer can be used. These are the results. This is a graph for the sugar estimation standard curve, and the rest three are for the honey, sugarcane juice, and palm jaggery. Here you can see the distillation setup. The honey was fermented for 14 days, and after that, the distillation was done as the foaming was stopped. The boiling temperature for this was 60 to 70 degrees Celsius, and the ethanol was approximately obtained 51 ml for the 400 ml fermentation medium. And for the palm jaggery, the boiling temperature was 60 to 70 degrees Celsius, and the ethanol obtained was approximately 40 ml. And for the sugarcane juice, the total ethanol obtained was exactly 50 ml. Here is the ethanol estimation done for the potassium dichromate by using the potassium dichromate titration. This table portrays the ethanol content determined by the titration and alcohol by volume of each substrate, honey, palm jaggery, and sugarcane juice. Here you can see the change in color of orange to blue due to the addition of ammonium ferrous sulfate drop by drop, indicating the presence of ethanol in the sample. This is the first test that was done to check the effectiveness of ethanol. The second test was flame test. Here you can see the blue flames coming. This indicates the presence of ethanol in the sample, and if it was orange, it would be methanol. This is the hand sanitizer that was formulated using the WHO guidelines. And to check the effectiveness of hand sanitizer, I did the palm test method, in which I used the agar plate method, in which you can see the left hand side plate here. The growth of bacteria is shown with my bare hands without the use of hand sanitizer. And after using the hand sanitizer, in the right image, you can see there is very less growth of bacteria. And I can say that's a big difference. Talking about the conclusion and the discussion, the palm jaggery yielded the least amount of ethanol compared to the sugarcane juice and honey, and the carbohydrate content of the original substrate was also estimated to determine the conversion efficiency of each substrate to ethanol. And the WHO recommendation formulation was used as per the guidelines. We can see the future research can explore cost effectiveness and large scale production using these substrates as well. And second and third hand generation feedstocks can also be used for the production of hand sanitizer. At last, I would like to express my sincere gratitude to Dr. Sanket Joshi, my professor, for his invaluable guidance and unwavering support throughout this project. His expertise and encouragement were influential in shaping this research, and I'm also thankful to laboratory staff at AIMT department for their assistance in providing the necessary resources that facilitated the execution of this study. Furthermore, I extend my heartfelt thanks to Dr. G.K. Aseri, Aparna Tripathi ma'am, Dr. Deepti Singh, and Dr. Geeta Sanket Joshi for their valuable insights and contributes them significantly enrich this project. Special appreciation goes to Dr. Sudarshan Singh Lakhawat, Professor AIB, whose assistance and collaboration were indispensable in various phases of my research. Their collective support and guidance have been crucial in the successful completion of my dissertation project.
Okay.
Thank you so much.
So, Urvi, it was a nice presentation. Now, I have a few questions like once the ethanol was produced, so what further processing you have performed to obtain the sanitizer?
Once the ethanol was produced, I firstly checked the effectiveness of it, if it is ethanol or methanol. So flame test was done and the other titration was done. And then I did the hand sanitizer formulation by WHO guidelines using the ethanol, glycerol, distilled water and hydrogen peroxide that was 3%.
Okay, and did you check the efficacy of the sanitizer? How it was?
Yes, I did. Yeah, I checked it using the palm test method.
One last question, like because ethanol production is an anaerobic process, you need not to provide the oxygen.
Yeah,
So
yeah.
what measure you have taken to ensure that the oxygen do not move in the flask?
I confined it in a dark place and like I obviously quarantined it for some time.
Okay, fine. So you may unshare the screen now. And next candidate is Asmita Singh. So Asmita, now you may share the screen.
Yes, sir. Sir, is it visible to you?
Yes, Ashmita, it's visible.
Okay, sir. So, good morning, everyone. Myself, Ashmita Singh. Here I am presenting my presentation.
So, my first question to you is, you have put the name of Ms. Tanvi Rathore. So, Tanvi Rathore is also working on this aspect.
Yes, sir, little bit.
So, I think that should not be there. And
Yes, sir.
please proceed.
Okay. So, my topic is morphological and molecular characterization in plants.
So, you are not properly audible. You can increase your volume.
Okay, sir. So now,
Yeah, be a little bit loud.
okay, sir. So my topic is morphology and molecular characterization in plants due to phosphate starvation. So here is, as we know that phosphate is highly important macronutrient which cannot be replaced by other elements such as liquid nitrogen or phosphorus or any other element, as it is very rare macronutrient in both soil and water. And it functions in lipid metabolism, signaling, as well as a structural component of nucleic acids and cell membranes.
You please also share your video. Ashmita, you can also share your video.
Okay, sir. So I'm not able to share it.
Okay, please continue.
A plant absorbs phosphorus in the form of inorganic phosphate from the soil, because if plant absorb the organic phosphate from the soil, then it absorbed in the form of phytic acid. And for the hydrolysis of phytic acids, the plants use phytase enzyme to release the phosphate from the phytic acid, which is mainly present in the seeds and present in very low amount in other part of the plant. So this is because of this effect, because of absence of this enzyme in other, or presence of very low quality of this enzyme in other part of the plants, the plant absorb phosphate in the form of inorganic phosphate. And plants may mostly receive phosphorus in the form of fertilizer, which are made up of rock phosphate. Fertilizer use is unsustainable, or it is very harmful for the plants or the soil, or the crops to use for a long period, because it can contaminate the soil. And when this contaminated soil move through to the water ecosystem, it also affects these water ecosystem, and it can cause water contamination, due to which it can create eutrophication. So it is very harmful for us to use a fertilizer in every form of fertilizer to provide the plants the phosphorus to complete their work. Then we have also studied how the phosphate affects the molecular and morphological characteristics in other plants, like as in Arabidopsis, it shows that when low phosphate is provided to the plant, the primary and lateral root the length of the primary root decreases, whereas the number of the lateral root increases, and also the elongation rate of the lateral root also increases. And in rice, it is found that these genes such as OSPX, PX1, PHR2, MIR399, and LTN1 are also involved in the phosphate starvation response. And it is found that the SPX act as a negative feedback regulator, which decrease the phosphate effect in the plants, and which when plant suffer from the phosphate starvation, it downstream its effect. And it is also found that hormones also play important role during the phosphate starvation conditions. For example,
Ashmita, you have only 5 minutes, so
okay,
you can move to your materials and methods and results.
Okay, so it is found that when the phosphate starvation occur, the root hair elongation increases, and PSI genes express, so that the PI uptake can also increase. All these methods help to increase the PI starvation, and these are the morphologies which are shown under the PI starvation condition. For example, in the roots, it is found that lateral root number increases, and the root hair number also increases, and the main primary root length decreases. In shoots, the secondary metabolites and the anthocyanin accumulation also increases. And sorry, this slide is not visible, sir.
So Ashmita, which gene is basically more important in regulating these kinds of phosphate stress, if you have any idea?
So basically there are so many genes involved in these, like PHR1 or SPX1, PHL or PTI, and these main genes are involved in the phosphate starvation, sir.
So, do you know about the regulatory process, how these genes are regulated?
Okay, sir. No, sir.
So,
Not in the detail.
so any idea how these PHRs are regulated? You have talked about SPX and all these things.
Yes, sir.
So I think all are involved in regulation of this phosphate starvation response.
Yes, sir.
So we have another question from Dr. Sudarshan.
Yes, sir.
So Asmita, I have one question that
Yes, sir.
in the plant cells, where is the phosphate most required?
Yes,
I
sir.
mean, what is the need of phosphate in the living cells?
Sir, actually, the phosphate is mainly used, as we know that in the DNA, it acts as a backbone for the DNA formation.
Exactly, it is required in the genetic material formation.
Yes, sir.
The ATP
And
formation also.
yes, sir. ATP is also
And-
also required.
And what do you think, like, what is or how the plant adapt to this starvation of the phosphate?
Sir, actually, when the phosphate starvation occurred, the plant part which is older, they move the phosphate stored in there that part to the new growing parts, and also they interact with the microorganisms such as mycorrhizals so that these bacteria can help them to absorb more and more phosphate from the soils. Also, they increase their secondary metabolism, and they decrease their photosynthesis, and they also increase the pi transports, and so these way they can also they increase their root hairs, so type they increase,
Okay,
so they
fine.
sustain
So
themselves from.
thank you very much. Now you can unshare the screen.
Okay, sir.
And we have the next participant, Miss Komal Veena. So Komal, now you can share the screen and start the presentation. Hello. Komal, are you there?
Yes, sir, I'm here.
Okay. So share the screen. Okay, so make it full screen now, and you can start.
Hello, sir. Good afternoon. Myself, Komal Meena. I am from MSc Applied Chemistry from MIT University, Rajasthan. I am here to presenting the presentation on eco-friendly synthesis of silver nanoparticle from natural extract and evaluation of antimicrobial activity. So, introduction. The rise of nanotechnology has revolutionized numerous fields, with silver nanoparticles emerging as one of the most widely studied due to their unique chemicals and physical properties. Silver nanoparticles are particularly valued for their potent and antimicrobial activity, making them highly useful in various applications including medicine, water treatment, and food preservations. Traditional methods for synthesizing silver nanoparticles often involve hazardous chemicals and high energy consumptions, raising concern about environmental and health impacts. In response, there has been
a growing interest in developing eco-friendly, sustainable methods for nanoparticle synthesis. Background on silver nanoparticles. Silver nanoparticles are particles of silver with size ranging from 1 nm to 100 nm. Due to their unique physical, chemical, and biological properties, they have become a focal point of research in various fields, such as medicine, electronics, and catalysis. Silver has long been known for its antimicrobial properties, which make silver nanoparticle highly effective against a wide range of microorganisms. Silver nanoparticles represent a powerful tool in the fight against microbial infections with applications spanning medicines, textile, water, and food preservations. Their broad spectrum activity combined with their ability to prevent microbial resistance makes them especially valuable in addressing challenges posed by multi-drug resistance pathogens. However, ongoing research is necessary to balance their antimicrobial efficacy with concern over toxicity and environmental sustainability. Synthesis process overview. To synthesize silver nanoparticle eco-friendly, start by preparing a plant extract, collect and dry plant material, then grind and extract it and boil water on ethyl or ethanol. Mix this extract with a 1 millimolar silver nitrate solution and heat it to facilitate nanoparticle formation, which is indicated by a color change. After synthesizes, characterize the silver nanoparticle using UV visual spectroscopy to confirm their presence and dynamic light scattering or transmission. Electron microscopy to check their size and shape. For antimicrobial evaluation, test and silver nanoparticle against bacteria or fungi using methods like the diffusion test and minimum inhibitory concentration test. Measure the inhibition or growth reduction to determine their effectiveness.
Okay, so Komal, can you directly move on the results? Yes.
Okay. The synthesized silver nanoparticle exhibit distinct color changes from yellow to brown color. And after from this to checking the actual nanoparticles are made from, we check from characterization like UV visual peaks, SAM, TEM for well-defined size and shape. The antimicrobial tests show that these nanoparticles effectively inhibit the growth of bacteria and fungi, highlighting their potential for various applications in medicine, water treatment, and beyond. Discussion. The green synthesis of silver nanoparticle using plant extract not only aligns with the sustainable practice but also provides nanoparticles with strong microbial properties. This method offers a promising approach for developing eco-friendly and antimicrobial agents suitable for various applications, including medical, environmental, and industrial uses. Conclusions. The green synthesis approach using plant extract not only provides a cost-effective and environmentally friendly method for producing silver nanoparticles, but also yields nanoparticles with significant antimicrobial properties. These methods offer a promising avenue for developing safe and effective antimicrobial solutions for a range of applications.
Okay, fine. So It was a nice presentation. Now, I have one question that you are saying the nanoparticles are toxic for microorganisms, isn't it? That's why they are stopping the growth of microorganisms, right?
Yes, sir.
So are they harmful for the human cells also?
Maybe because I think
You got my question. I am asking like, are nanoparticles harmful for the human cells if they are harmful for the microorganisms? Okay, fine. So you may unshare the screen.
okay, sir.
And let's start with the next presentation. Shasta Saeed Al-Kindi. So are you there?
Ms. Shasta Saeed Al-Kindi, are you there?
Excuse me, sir. Sorry to interrupting. Can I leave the session?
Yes, you can.
Okay, sir. Thank you.
Ms. Shasta Saeed, are you there? Okay, we'll next move to the next speaker. So,
Hauda Saeed Murshid.
Hauda Saeed Murshid. Hauda Saeed Murshid, are you there? So, next speaker is Gandhi Bhaskar Patradulamka. Mr. Gandhi Bhaskar, are you there? Ms. Krati Vyas, are you there?
Yes, sir.
So, Krati, can you please share your screen and proceed with your presentation?
Yes, sir. So my screen is visible.
Yes. So now you can make it full screen. We'll start the slideshow.
Is it visible?
You can make it full screen. It is not full screen right now.
Sir, I've already done. Is it now visible in full screen?
If you are not able to make it full screen, then you can proceed in this format only. Okay, you need not to make it full screen if you are not able to do it. Okay, please
Okay,
proceed.
sir. Okay, sir.
Okay, you may
So,
start.
good afternoon, everyone. Myself, Krati Vyas. I have done I've graduated from ICGETEI Jaipur itself. So, as everybody know, there is an increase in antibiotic resistance is becoming a major issue in the healthcare industry. So, my topic is on isolation of novel microbes which produce antimicrobial agents. So the objective of this study is to isolate the novel bacteria which produce antimicrobial agents. So the isolation of bacteria is done from different soil samples which were collected from different places. So the discovery of novel bacteria can have a new action of antibiotics mechanism against the pathogens which have gained resistance against the current antibiotics, and it can also broaden up the arsenal of antibiotics present in current situation. Some of the bacteria which were discovered and are used as antimicrobial agents against pathogens are Actinomycetes, Bacillus species, which is Bacillus subtilis and Bacillus
But
coagulans.
your slides are not changing. So please move on the next slide.
Okay, Pseudomonas species and Lactobacillus species. So these were the materials which I used in the entire research. These were the methods which were used: crowded plating method for isolation, spreading for further isolation, and after that antimicrobial resistance test and competitive innovation test was
Hello,
performed.
your slide is
Yes.
not changing, please. You are still on the first slide for us. Now it's changing. Okay. Yes, you may proceed.
So methods were followed. Antimicrobial resistance or competitive inhibition test was followed by zone of inhibition test, and after that identification tests were performed. So in crowded plating method, it is a method which was used during the isolation of all soil samples. So in this, I have taken 10 gram of my soil sample in 100 ml of distilled water and kept it in water bath at 80 degrees Celsius centigrade for 30 minutes for heat fixation. And after that, I have done inoculation in which up to 6 dilutions were prepared with 9:1 ratio, and that sample is spreaded evenly and carefully on the plate. And for incubation, I have kept it overnight for 37 degrees at 37 degrees Celsius. And after that, the colonies was observed. And after that, I have performed antimicrobial resistance or competitive inhibition test, in which the colonies which were observed was further isolated as the counter strain, in which C1 was obtained from the sample, and it was used as the counter against the all 19 isolates to check the antimicrobial resistance. After that, C2 was similarly C2 was used as counter against all the isolates which were obtained. And after that, I've used one Pseudomonas strain against all the isolates, one Candida strain against all isolates, and one more C13, which was also obtained from the sample, was used as counter against all the 19 isolates. After that, I've performed zone of inhibition test for two of my samples, which was C13 strain and C14, because C13 strain was showing competition against the C14. So in this TSA plates was prepared, in which after that, for inoculation, C13 was which is antibiotic antibodies producing, was inoculated in a flask of TSB, and the inhibited, which was C14, was inoculated in the other flask and kept for 24 hours at 37 degrees Celsius. And then after 24 hours, this was taken out, and C13, which is antibody producing, was centrifuged at 1400 RPM for 10 minutes. And after that, during that, I have spreaded my inhibited bacteria on the plate. After that, I've done the antimicrobial application, in which after the plate dried, I made 4 wells, 2 vertical and 2 horizontal, in which vertical I've filled with positive control, and on negative horizontal I've filled with negative control. So after that, the identification tests were performed, which were gram staining, negative staining, and endospore staining. So the results for of the isolation are these. One was neem soil sample, which was from Amity itself. These two are 10-4 and 10-5 isolates, which were showing different colonies. Other was dumping yard soil sample from Amity itself. These two were showing the different colonies. And last was the Saras fade and DAF liquid samples, which were used, but these were not showing different colonies. These were showing similar colonies. So after that, this is the competitive inhibition test which I have performed, in which C1 was the counter and the other samples was streaked against it, but it was compatible with all. Similar with C2, it was also compatible with all, and Pseudomonas was also compatible.
Candida was also compatible with all, but C13 was the only strain which was not compatible with C14. It was blocking its growth. So I have isolated C13 against one which was C14 isolate. And after that, I performed the zone of inhibition test of C13 and C14. So one was centrifuged and other was non-centrifuged, and was showing a clear zone and inhibiting the growth of C14. In the identification test, gram staining, both C13 and C14 are rod-shaped bacilli, gram positive and purple colored. In endospore, they have endospore present both, and in negative staining, they both were rod-shaped. So the discussion, I made an attempt to isolate a novel bacteria or unidentified bacteria which produce antimicrobial agents from different soil samples from different areas where there can be a possibility of their presence. So with this attempt, I have isolated one such bacteria which is producing antimicrobial agents among all the 19 isolates, which was C13, and this bacteria was isolated from the sample of garbage dumping yard soil of Amity, University, Rajasthan. But for future recommendation, there are more identification tests which is required, like metagenomic approaches can be taken to analyze the microbial community. High throughput screening can be performed to implement identify the antimicrobial producing microbe, and genome mining can also be performed to identify the if the bacteria is novel or not. In acknowledgement, I would like to express my sincere gratitude to Dr. Sanket Joshi, who was also my guide throughout my research project, and with his expertise and encouragement were the instrumental in shaping this research. I'm also deeply thankful to the laboratory staff at AIMT for their assistance and for providing the necessary resources. Furthermore, I extend my heartfelt thanks to Dr. G.K. Seri, Dr. Aparna Tripathi, Dr. Deepti Singh, and Dr. Geeta Joshi for their valuable insights and contribution that significantly enriched this project. Thank you.
Okay, Krati, very nice presentation. So I have few queries from you. So basically, when you started your work, when you're trying to isolate these different bacteria from different sources, so what media you utilize in the beginning for screening purpose?
I can't. You're not audible to me.
I'm not audible? Okay.
Yes, sir.
Am I audible now?
Yes, sir. Now.
So I'm asking, when you started your studies, in the beginning, which media was utilized for screening of different bacteria from different sources, basically?
Sir, soya KC Nagar was used.
Okay. And how many isolates were there in that first screening?
Sir, 19 isolates.
So only 19.
Yes, sir.
So I saw one of your plate, it was showing a very large community of bacteria growing there in initial studies. So like you can see, all the plates are full.
Yes, sir.
So how can you say that only 19 isolates were there? So there can be many more as well.
Yes, sir, but sir, I have only isolated those who were showing competition against the others.
Were showing?
Which were growing more.
Which were showing antimicrobial properties?
Yes, sir.
So, but I think you have studied then all the isolates for this kind of study. Maybe you have 100 isolates, you have plated all of these on that on specific plates, and then you have concluded, okay, this is isolate which is having antimicrobial property. So maybe you have utilized many isolates initially.
Yes, sir.
Is it so? So how many isolates in the beginning you have used? You're saying only 19, so why not others?
So because they were same.
Maybe you have used some kind of criteria like color or something, or gram staining in the initial phase, or directly you plated against basically it's
So
tried.
directly.
Okay,
Directly.
fine. And now, how many isolates are you basically maintaining?
So only two, C13 and C14.
C13 and C14. Okay, and what are your future plans with these isolates?
So if I get any chance, I want to go for further identification if is the bacteria is novel or not, to identify that.
So are you going for whole genome sequencing or just identification of that bacteria?
Identification.
Okay, fine. Thank you.
Okay.
You will
Thank you,
leave
sir.
now. Please unshare your screen. So next speaker is Ms. Kritika Sharma. Ms. Kritika, are you there? Are you listening to me?
Yeah. Hello.
Yeah, Kritika. Please share your
Yeah.
screen.
Am I audible, sir?
Yes, you are audible.
Okay. Just a moment. So is my screen is showing to you?
Yes, it is visible, but you can move to the slideshow mode.
Okay, the full
Full screen,
Yeah.
right?
Just a moment. Is it fine now?
Yes. Now you may start.
Okay. So a very good afternoon to everyone. Myself Kritika Sharma, and I'm a research scholar from Amity School of Communication. And I had written a paper with the authors with Dr. Pallavi Mishra, Dr. Shweta Kulshrestha, and Dr. Tanushree Mukherjee. So the title of my paper is A Bibliometric Analysis of Nanoparticle Research in Fruit Juices, that is Strengths, Innovations, and Applications. So we begin with the introduction. The application of nanoparticles in fruit juices signifies a very significant breakthrough in the field of food sciences, that is with the objective of enhancing nutritional value, safety, and sensory attributes. So with the we can talk about nanotechnology. The vitamins, antioxidants, and various bioactive compounds can be easily encapsulated within the nanoparticles, which further improves their stability and bioavailability. So for example, we can have nano encapsulation serves to safeguard, that is sensitive nutrients from degradation during processing and the storage. So somewhere ensuring that the consumer obtain the maximum health benefits. Moreover, nanoparticles can be also utilized to develop more effective preservatives, which is prolong the shelf life of fruit juices by inhibiting microbial growth and preventing oxidation. So the integration of such nanoparticles can be censored through nanosensors, that is into fruit juices packaging, facilitate real-time monitoring of product quality and safety. So the sensors can easily detect contamination, spoilage, and alteration in the chemical composition of the juice, thereby providing consumer with an additional layer of assurance. However, despite some of the potential advantages, the incorporation of nanoparticles in food and beverages raises significant safety and regulatory issues. So the long-term health implications of nanoparticle consumption still remain inadequately understood, highlighting the need of comprehensive research and regulatory scrutiny to ensure the consumer safety. So the research questions of this particular research paper are, so the first research question is, has there been a significant change in the number of publications on nanotechnology and fruit juices from 2014 to 2024? The second one is, what are the main documents explored in the research on the nanotechnology and fruit juices after the implementation? The third one is, which researcher and institute have been the most productive and in terms of publishing on this topic? The fourth is, hello.
Yeah, yes, you can continue.
Okay. So the fourth research question is which country has received the highest number of paper and citations in the field of nanotechnology and fruit juices? So when we talk about the biometric analysis, it is basically in a health field. We can say that bibliometric analysis are mostly used to measure the influence or impact of any research articles. The method, sometimes estimates how much influence or impact of a selected research article has on the future research papers. It usually sometimes does this counting by the number of times the article is being cited after its publication. So the research methodology and analysis is related to that. I have using the framework from 2014 to 2024. The bibliometric analysis has been done by studying this particular time limit papers, and the analysis aims to identify key research themes and influential entities in the field. By reviewing all the literature on nanotechnology and fruit juices, a significant amount of research has been found in this paper. And the analysis will also consider variables such as citations, publications, journals, authors, and countries. And additionally, an examination of keywords which is indexed in the articles will be conducted to identify research groups and understand the research themes associated with the nanotechnology and fruit juices. So here we can see that the document by year, the influential documents has been covered by the 2024. And if we talk about the document by some subject areas, so the most highest subject area which is comes under the other subjects, and another one is the biochemistry, which has a large number of articles published related to nanotechnology and juices. And then on agriculture subject area, then chemical, and then chemical engine, that is 10.0%. And when we talk about the document by type, we have found that articles have been more improvement for the publishing material on nanotechnology and juices. And rest, we have review articles. In the last, we have very least number, that is the editorial. Hello?
Yeah, I am saying like we have 5 minutes limit only, so please conclude quickly.
Yes, sir. I'm about to just come online. So the authors which I have cited are most articles, that is ICBAS, HMN, that is compared with the 15 document counts of 15 authors. And this is somewhere we have used VS Viewer for this. And we can find that India has the largest number of publishing articles, that is in the barrel, that is China, Mexico, and Pakistan and all. So India has the largest number of articles. So by the document per sources, we have counted from up to 10 sources, and most of the articles has been published by 2021 to 2024 in under the International Journal of Biological Micromolecules. So the interpretation of my study is the analysis delves into the interact web of scholarly endeavors by curating metadata from retrieved publications. And it also uses the key terms like nanotechnology and fruit juice, direct the investigation in the field. The highlights of essential elements such as publication titles, author attribution, publication years, journal, and keywords. This comprehensive compilation of bibliographic data lays the groundwork for future analytical endeavor. And the analysis has been done on the street.
Fine, yes, Kritika. We go to your views. Basically, we have very limited time here. So I have one question from you. Again, the same question. Basically, you have used only 2 terms, nanotechnology and fruit juice, for basically finding your different articles. So you haven't used, basically you can use more terms to find out more bibliography in this area. So how can you say that this is the only, these are the only references which are present there? Because if you will basically diversify your keywords, then you will find more articles as well.
True, very true, sir. Actually, I have just indulged myself with only these 2 keywords. On that parameter, I'm saying these are the results. But yes, you're very true in that. If I'm using a lot of keywords with this, there are more and more research articles I can found it and make it more progressive and the future for the future endeavor.
Yeah, that's what what I'm saying, that it
Yeah.
will make more comprehensive. We will be using
Yes,
more
sir.
terms. Okay. So
Yes.
this only using these two terms, making it a limited study. Okay.
Limited study. Yes, sir. I'll try to make it more integrated with so many keywords in it.
Okay, fine, Kritika.
Okay.
You will leave now. So we you please unshare your screen.
Yes, sir.
So next speaker we have Miss Asmita Mukundrao. Asmita, are you there?
Excuse me, sir.
Yes, please.
So, sir, can I leave that portal?
Yes, you can leave.
Okay.
You can leave.
Okay. Thank you so much.
Asmita Mukundrao
Tina Patidar.
or Tina Patidar, are you there? Tina, can you join?
I will share.
Yes, please share your screen. Miss Tina? Miss Tina, are you sharing your screen? So I think we have some, Miss Tina have some issue in joining the link. So we may call our next speaker, who is Gandhi Bhaskar Patradu Lanka. Mr. Lanka, are you there? Mr. Gandhi Bhaskar. So I'm announcing the name of the speakers who are remaining. So if any one of you are present there, please join the link. So first one is Shatha Said Al-Kindi. Shatha Said Al-Kindi.
Sir, hello.
Yeah, who is there?
Sir, this is Mamta, Racharla Mamta.
What's your name?
Mamata, sir.
Mamta.
Yeah,
Mamta.
yes, sir.
We don't have your name in the list.
Mamta?
Yes, sir. Yes, ma'am.
Mamta, have you registered for the author link, through the author link?
I have registered through that, ma'am.
Have you submitted the abstract?
Yes, ma'am.
Okay, and when did you register? Have you registered
No,
online or offline?
Online, madam. I have registered for online.
Which date you had registered?
From Telangana.
When?
I'm from Telangana. I'm from Telangana, madam.
No, you are from Telangana, but which date do you register? Do you have done the registration for the?
Registration on Monday, madam. On Monday, I have registered.
Tina, are you able to hear me? Tina, please share your screen.
We cannot hear you. So
Please unmute yourself. Yes, your screen is visible, but your voice is not coming.
I think you are muted. So either you unmute yourself.
Yes, sir. Am I audible, sir?
Yes.
Yes. Sir, good afternoon, sir. My name is Tina Patidar. My presentation topic is isolation, identification, and quantification of quercetin from plant. The aim or the abstract of the research is as the Nicanthes arbutus is having a many Ayurvedic properties or medicinal properties which receive a significant attention due to its pharmacological and biological characteristics. This study aimed to develop a fingerprinting profile of ethanolic leaf extract of Nicanthes arbutus using SPTLC, a technique for separating identifying compound in a sample, and the ethanol used as a solvent. These are the objective of the research, that is to perform the extraction of leaves to generate a qualitative fingerprinting of ethanolic extract and to quantify the quercetin in Nicanthes arbutus extract. The experimental part in which we have first performed the maceration for the action of the leaves. So powder the leaves of first we allowed to dry the leaves in shade, and it get grounded, and the maceration is done by macerating the leaves in for the 24 hours in ethanol, and the extract is collected and evaporated by rotary vacuum evaporator. So first for the SPTLC, we prepare the standard solution of quercetin. So for this, that we dissolve the 10 mg of quercetin standard in 10 ml of ethanol, and prepare the standard, and this standard is auto applied by applicator in HPTLC in concentration range of 0.4, 0.6, 0.8, and 1 and 1.2 microliter. For the performing the HPTLC of ethanolic extract, we also take 10 mg of ethanol, dried ethanolic extract, and dissolve in 10 ml of ethanol, and we applied 0.6 and 0.8 microliter solution of that extract solution. The temperature is 85 degrees Celsius, and which we perform for 5 minutes, and wavelength is 254 and 366 nanometer. The HPTLC mobile is toluene, acetone, and formic acid in ratio of 9:9:2. The saturation time is 20 minutes and drying time is 5 minutes. These are the SOP which we have prepared in for HPTLC. So first 5 is for reference standard, and 6 and 7 is for sample, that is extract. The result of this is we found that the R value of standard is matching R value of extract of ethanolic in the Nicanthes arbutus extract is matching to standard, and these are the chromatogram which we obtained after the analysis. By this chromatogram, we quantify the quercetin in extract, that is 705.5 nanogram per ml, and in 0.8 microliter, we have calculated that is 914.8 nanogram per ml. So the conclusion is we perform the HPLC of Nicanthes arbuticid. This information could be considered as a standard, which is taken as a reference for quality control purpose. And moreover, the therapeutic potential of quercetin was demonstrated in chromatographic experiment using a leaf extract on Nicanthes arbuticid. So this can be a useful method for the quality control purpose in other plant extract for quercetin. These are the references. Thank you.
So, Ms. Tina, so I have few queries. To you.
Yes.
So, what is this plant? Nicanthes arbuticus?
Arbuticus.
What is common name?
The common name is Harshingar, sir. Harshingar.
So, why only this plant? My first question is why only this plant for isolation of quercetin?
Sir, I'm doing the PhD PhD on this plant for the anti-cancer activity. So I have performed the HPLC of this plant. So I have chosen this on the basis of screening of different plant, which all having anti-cancer activities. And this is the plant.
Screening in terms of what? You screen them.
Screening in of first is the anti-cancer activities, and other is the plant parts is so sir explore अभी इसका research थोड़ा कम हुआ है तो
okay, fine.
Looking for.
Fine, Tina. I have another question. So you have used leaf extracts here. So so maybe that plant is having different types of leaves. Some are young, some are older ones, some are withered one. So have you used
yes, sir.
specific kinds of leaves or have pulled them out?
Sir, older leaves, fully grown.
Fully grown leaves. Okay.
Yes.
Okay. Then and mobile phase which you have used. So only
Yes, sir.
tried one mobile phase for HPLC or tried different combinations as well?
Sir, we have tried for other mobile phase also, but spot for quercetin. So we have also tried for toluene acetic acid, ethyl acetate, and toluene ethyl acetate, ethanol.
Okay, fine, Tina, fine. Thank you, thanks a lot. You may leave now. You may unshare your screen. So, we're calling on our next speaker, Mr. Gandhi Bhaskar, Pachadulanka. Are you, have you joined? Mr. Gandhi Bhaskar? Okay, Mr. Shada Saeed, are you available? Ms. Shada Saeed? Hauda Saeed Murshid? Mr. Hauda Saeed Murshid?
Yes, I'm here.
Okay. Ms. Murshid, can you please share your screen? Yeah, it's visible now. You can proceed with your presentation.
Okay, good afternoon, everyone. Thank you for being here today. My name is Huda from Oil and Gas Research Center, Sultan Qaboos University.
Please be a little louder, okay?
Okay. Today I will be discussing metal oxide nanoparticle for enhanced oil recovery application. In my presentation, first I will talk about introduction, then literature review, methodology, result and discussion, and then conclusion. Nanoparticles are increasingly being used in enhanced oil recovery to improve the efficiency of extracting oil from reservoir Their small size gives them a unique property, making them variable in various EUR techniques through improving mobility control, changing the wettability, reducing the interfacial tension, and enhance foam stability. The optimal use and long-term effect of these nanoparticles in EUR need further investigation to ensure their efficiency and economic viability. So our objective was to investigate the effect of silicon dioxide and titanium dioxide and mixture of both silicon dioxide and titanium dioxide in improving recovery factor. Many investigations have reported the promising result in increasing oil recovery by injecting nanofluid due to increasing the viscosity of nanofluid, decreasing interfacial tension, and changing the wettability. For example, using titanium lead to increase the oil recovery by 41% to 55%. Another study used silica, which has increased oil recovery by 4% to 10%. Then the methodology, in our research, we use crude oil with 23 cb viscosity and brine 1% NaCl, and we use nanoparticles as silicon dioxide and titanium dioxide as transparent water dispersion solution from research nanomaterial company. And we use Beria sandstone core with permeability of 200 millidarcy, and we use glass slabs for contact angle measurement. And here is the setup for core flooding experiment and the contact angle measurement setup. We did the experiment and contact angle measurement at 50 degrees Celsius. Here is the result of our project. The first graph illustrates the recovery factor with pore volume injected for silica dioxide. The recovery factor increased to around 30% with injection of the brine with different concentration of silicon dioxide from 0.05 to 0.2% are injected. The recovery factor increased to 44% and get 14% extra oil recovery. The second graph, the titanium dioxide, follow the similar trend with a sharp initial increase in the recovery during brine injection to reach 68% of the oil, and with different concentration of titanium dioxide from 0.05% to 0.2% show improvement in the oil recovery factor by around 15% extra oil recovery to reach around 83. For the third graph, which illustrates the titanium dioxide with silicon dioxide mixture, the brine after brine we get around 65% from the oil, and after injecting different concentration of titanium dioxide we reach a recovery of 80%, which around 16 extra oil recovery, and this is the highest among the others. This graph illustrates the contact angle measurement for three systems, silicon dioxide and titanium dioxide, and the mixture of titanium and silicon dioxide. The silicon dioxide and titanium dioxide mixture exhibit the lowest contact angle. It reduces the contact angle from 138 degree in brine to 80 degree. For high concentration, the drop was flat. This is a clear indication that the surface becomes so water wet at this concentration that the contact angle approaches zero. And no contact angle can be measured. So to conclude, this study highlights the effective of titanium dioxide and silicon dioxide nanoparticle and enhanced oil recovery by changing the wettability, And the cool flaring experiment demonstrates that the addition of titanium dioxide and silicon dioxide nanoparticle to brine significantly improved the recovery factor, with the highest recovery, nearly 80, achieved at concentration of 0.2. And these core flooding results are consistent with the contact angle finding. Overall, both the contact angle and core flaring result confirm that the use of titanium and silicon dioxide nanoparticle can optimize oil recovery by changing the wettability. Thank you.
Okay, thank you, Ms. Murshid. A nice presentation, nice piece of work. So I have one basically you have used two different kinds of nanoparticles, and one is basically combination of these two particles. So is this the only nanoparticles which can be effective, or you have basically what is your criteria of choosing only these nanoparticles and these elements?
I worked before with many nanoparticles like aluminum oxide, iron oxide, many particles, and then I choose the best titanium and the silicon. I repeat the experiments and I mix them together to see the effect.
Okay, fine. So you have tested others as well.
Yeah.
Okay, and then the next question is basically whether these can be used for your crude oil or as well as some refined oils as well.
I tested for crude oil.
For crude oil.
Yes.
Okay, thank you, Ms. Murshid. Nice presentation.
Thank you very much.
Right, thank you. So you may leave now. You can unshare your screen.
Okay.
So can we have our next speaker with us, Ms. Shatha Said Al-Kindi? Ms. Shatha Said Al-Kindi. Mr. Gandhi Bhaskar. Mr. Bhaskar?
Sir, am I audible, sir? Hello.
Yes, you are pretty much audible. You please share your presentation. Are we audible to you?
Actually, I'm getting very little audio from your side. Anyway, I'll go ahead with the presentation steps. So
Sorry,
can you share the PPT from your side, sir? Or
I don't have your PPT.
So how do I share my screen, sir, from here?
There must be some option on your side. It is written on the bottom side. You can see it is written as share screen. Or either you share it to Deep sir, so that yes, now it is visible.
It is visible. Please.
Is it visible, sir, now?
Visible, but please zoom out your slides. The whole slide is not visible. Yes, now it's visible.
Okay, you can start.
Sir, my screen is visible, sir?
It is visible. You can quickly
start. I'll go ahead, sir. I'll go ahead.
You can start now. We have 5 minutes only, so please quickly conclude.
So today my topic of presentation is the effect of concentrative meditation on cognitive functions. This is a series of controlled experimental studies done on attention, processing speed, short-term memory, cognitive flexibility, and impulse control. So concentrative meditation is focusing on a specific object, thought, or activity. The techniques that were used are Trataka, gazing meditation, and Anapanasati, mindful breathing. Cognitive functions that were studied were attention, processing speed, memory, flexibility, and impulse control. So the aim of the study is to find out the effect of cognitive meditation on various cognitive domains in healthy individuals. This study investigates the immediate effect of Trataka meditation on processing speed and short-term memory, and long-term effects of Anapanasati meditation on processing speed, and immediate effects of extended meditation practice of 2 hours on cognitive flexibility and impulse control. 121 healthy individuals were participated in the study. They were divided into control and meditation groups. 1 second, please. Sir, मैं आ रहा हूँ सर. Yes, sir, आ रहा हूँ सर. Sorry for the distance. So five separate experiments were conducted focusing on various cognitive measures. Methodology: experiments on both short-term meditation and long-term meditation were done. Short-term meditation consisted of 15 minutes of practice, and long-term consisted of 6 months of meditation practice. Both between subject and within subject designs were used. Results: in the experiment 1, the study on the effect of processing speed, 15 minutes of Trataka meditation improved did not significantly improve the reaction time compared to the control group. These were the tables showing no significant improvement in the reaction time. Then the experiment 2 shows the immediate impact of on short-term memory. So 70 participants were taken. and 15 minutes of Trataka meditation was done. And it showed a non-significant improvement in short-term memory in the meditation group. So this shows that even 15 minutes of Trataka meditation can have some effect on the memory. These are the tables showing the improvements, the P scores, P values. In the experiment 3, they studied on long-term effect of meditation on processing speed. 20 participants did long-term meditation of at least 6 months. Anapanasati meditation they were doing this Anapanasati meditation, and the reaction time was used to assess the processing speed. Long-term meditation significantly improved the reaction time, and this implies that there is a significant positive effect on the post-processing speed. So these are the tables showing the improvement in the reaction time between meditators and non-meditators, long-term meditators and non-meditators. So this is a graph showing the difference. In the experiment 4 was done to study the effect on cognitive flexibility. Extended meditation session of 2 hours plus meditation single session with multiple testing was done to study the cognitive flexibility. The results showed significant improvement in the normal and interference reaction times, proving that extended meditation practice of meditation practice can enhance the cognitive flexibility. These are the tables. So this graph shows that as the person, the same person did multiple days of meditation, extended meditation, on day 1, day 2, day 3, by the day 6, there is a significant improvement. Also with the stroke interference, also there is a significant improvement by day 6. Impulse control. The experiment 5 is dealing with the impulse control. Again, this shows that extended meditation practice will has improved the impulse control as tested by the go-no-go task. This go-no-go task tests the ability to control impulses and also attention. So this has significantly improved with the extended meditation. These are the tables, and this is the showing the significant improvement on meditation days. There is a significant improvement. Day 1, 2, 3 are without meditation, and day 4, 5, 6 are with meditations. So there is a significant difference between the days without meditation, on the days with meditation. So discussion. This shows that both short-term meditation and long-term meditation has impact on cognitive functions. So this series of 5 experiments shows that meditation affects the various cognitive functions such as processing speed, cognitive flexibility, executive functions, and impulse control. So further studies should be done with larger study subjects. Thank you.
Yes, okay, so it was a nice presentation. I have a few questions like, first of all, what technique was used to measure the STM? Hello, can you hear me? Hello? Bhaskar, can you hear me? चलो ठीक है
कॉल कर लो. He is visible. Hello.
Sir, my presentation was audible, no, sir, clearly?
But we have some questions, sir. You can ask.
Okay, so I have some questions regarding the presentation.
Yes, sir.
Yeah, I want to ask like what technique you have used to measure the memory in this case?
Memory word list, sir. Word list was used.
Okay, and can you tell me which part of the brain is associated basically with memory?
Memory, there are multiple regions are involved, sir, like cortex, hippocampus.
Okay, so you have used fMRI or not? fMRI technique?
No, sir, this is only objective, subjective assessment only, without any objective fMRI or anything.
Okay.
Only on the word list test, sir.
And how many subjects you have selected in the study?
So five experiments are there, sir. First experiment, in the experiment 130 participants, sir.
30 participants. Okay, fine. Thank you.
Thank you, sir.
Okay, so you may unshare the screen and yeah, so